Good afternoon, and thanks everyone for joining our panel. I'm Geulah Livshits, a biotech analyst at Chardan. So the last several years have seen a number of regulatory approvals for CAR-Ts and gene-modified cell therapies, and coupled with efforts to apply these modalities in new indications. And in parallel, we've seen innovation around the types of cells that are being developed as therapeutics, as well as advances in manufacturing processes. And manufacturing is really central to the success of cell therapies, where the process can define the product as a living drug. The way you source, culture, and modify cells can drastically impact the composition and the activity of the final product. So in this panel, we'll convene representatives of companies advancing different types of cell therapy modalities to understand the important considerations for feasibility in the clinical as well as in the commercial setting. So I'm joined by, in alphabetical order, Dr. Ed Samuel, EVP Technical Operations of Achilles Therapeutics, Dr. Kinnari Patel, President COO of Rocket Pharma, and Dr. Armon Sharei, Founder and CEO of SQZ Biotech. So the format for this session is a roughly 45-minute panel discussion. And again, if any of our audience attendees have questions, feel free to type them into the question box under the video player, and we'll try to ask them on your behalf. So to begin with our group, I'd ask our panelists to briefly introduce themselves, and we can start with Kinnari, Ed, and then Armon.
Hi, thank you so much for having me here. I'm Kinnari Patel, President and CEO of Rocket. I've been with Rocket Pharmaceuticals for about 6 years. And one of the beautiful things about Rocket is we're focused on something that's near and dear to my lifelong work, which is developing therapies for rare diseases. At Rocket, we have both ex vivo lenti and AAV based in vivo therapies that are available for pediatric rare diseases. What's unique about Rocket's philosophy is we try to be first and only in class in all of our indications, and we try to think about product approval before we even submit an IND. So always thinking about the end game in mind, given that our co-founders and our initial management team are really drug developers. And I think that has enabled us to really lead for the programs that we have in the pipeline to phase 1 clinical studies, as well as phase 2 programs nearing top-line data readout and filing activities. From cell and gene therapy manufacturing perspective, our ex vivo lenti programs are all outsourced through CDMO, and our AAV manufacturing is brought in-house. So we have a little over 100,000 square feet of in-house manufacturing that not only has commercial grade manufacturing capabilities for EMA and FDA, but we have QC process development to allow for future expansion of our pipeline. And that makes it really kind of interesting and challenging, which I'm looking forward to this conversation to talk about those pros and cons of outsourcing, insourcing, and different technologies and how we can apply synergies across the board.
Hi, Ed Samuel, so Executive Vice President at Achilles Therapeutics. Been with Achilles for about 4.5 years, but spent my entire career, the kind of last 20 years, working in the field of cell and gene therapies prior to them being considered in medicines. So very much starting out with an academic background and then transitioning over to industry over the last kind of 6 or 7 years. And at Achilles, we have 2 first-in-human studies, phase 1, phase 2 in the clinic, in lung cancer and in metastatic melanoma, where we're treating patients with our somatic cell therapy targeting clonal neoantigens.
And I'm Armon Sharei, founder CEO of SQZ. We spun out the company from MIT based on the discovery of a new way to deliver materials into cells, where essentially what we found is that if you take cells and squish them at high speeds, you can disrupt the membrane temporarily such that anything that's outside will go in, and then the cells will just detect that disruption and close it back up. And while this solves in some ways a very esoteric problem of how do you put materials into cells, it's actually pretty fundamental to how you can ultimately engineer cells to do all the different things you would want, and how can you implement them in a relatively scalable and accessible way for the patient. So with SQZ, we felt that we could overcome a lot of the underlying issues that have constrained what the cell therapy field can do right now when it comes to what biologies are accessible and what applications you can feasibly implement this for. So we've been exploring a lot of different therapeutics with this, and our initial focus is all around trying to direct the immune system, so engineering functions like antigen presentation, and both in the context of activation of T cell responses as well as tolerization of T cell responses, to go into areas like oncology, autoimmunity, and infectious disease. We have 3 active trials on the oncology side, all of which have readouts coming later this year, and we have a major partnership with Roche as part of one of those programs. And on the manufacturing side with Squeeze, because it's a very different way to implement cell therapies, all of our manufacturing so far has been under 24 hours for our patients. Vein to vein is about a week. We don't need to precondition the patients. They don't need to get hospitalized on the other end. So it's already demonstrating just how different a cell therapy can be from a patient perspective. And we are moving towards a point of care implementation of the system, meaning that we integrate everything into a single unit that could ultimately sit at the hospital site without the need for a clean room, and therefore actually just produce your cell therapies on site in a much more distributed model. So excited to talk about all those different areas as part of the panel today.
Great. So let's start with the positives. I think cell therapy as a field, as I mentioned earlier, has been evolving for some time. From each of your perspectives, what are the important manufacturing-related advances that have gotten the field to where it is today? And let's go in the same order, Kinnari, Ed, and then Armon. You're on mute, I think.
That helps. Muting also helps. When I think about the prospect of advancements in innovation, I think especially in the last 5 to 6 years, there have been a lot of advances that have happened. And what it has enabled for us is to think about the disease and keep the disease focus first and foremost. So the way at Rocket we think about things is if you think about a disease and the program-specific challenge or cell of interest, then you can thoughtfully develop therapies or use a platform to apply directly in a mechanism of action that targets a cell of interest. So I think that's kind of been the more advancements we have, the more we can do this more specifically and individualized for diseases and patients' needs. And I think that's been really cool. In terms of other things that I've seen advance and, going from qPCR to ddPCR is really automation when it comes to analytics. And how do you think about analytics, standardization, and where it makes sense, how do you do cell counting and scalability? And the third thing that I think is really critical is the collaboration and knowledge sharing that has happened in different forms like this across the companies have allowed us to really implement some of the advances when it comes to ex vivo lenti, vector design, as well as AAV scalability. And those are all the things that while on one hand, I feel like there are a lot of advances made in the last 5, 6 years, I'm really excited about the next 10 years of how do we do this even more efficiently and more scalably.
And you're also muted.
We're pretty good at this, aren't we? Yeah, from my perspective, I think, focusing on specifically on Achilles, given that we're manufacturing precision medicines, 5 or 6 years ago, this, our platform probably wouldn't have been possible given where sequencing was. And we've made rapid advances in terms of the turnaround time to sequence a patient's whole kind of genome. And the advances as well with kind of AI and our bioinformatics platform. So, that's been a kind of a rapid maturation in the field that's enabled us to kind of bring our technology into the clinic. I still think as a whole, the field of cell and gene therapy, I wouldn't say has suffered from the lack of technology available, but it's only really beginning to catch up. I think the investments into the clinical development came early, and the kind of biotech companies and the kind of technology that was being spun out of academic facilities and units. And now we're really just only beginning to understand what are the bottlenecks for getting our therapies at scale to patients. And so I think we're seeing lots of exciting development with new technology. I think more and more developers are looking at closed systems. Again, academic manufacturing has often been quite labor-intensive, open processing. And I think a lot of the developers in the technology space, whether it's equipment, in the GMP environment, are thinking very early about what's required from a GMP perspective. So, doing that right from the inception rather than trying to find a space and then retrofitting equipment because you see the opportunity in the GMP space. So thinking about GMP earlier is definitely, something that we're seeing at Achilles much more. And I think that the last thing that we're seeing that's helping bring forward the manufacturing is just, I guess, growing out the skills base as well. And that's something really that has not been, hugely available to all of the biotech companies and kind of developing their therapies is just, the pool of talent to pick from. And so I think developing that is enabling us to get our technologies and our therapies to patients quicker.
Yeah, and I think just to build on what Kinnari and Ed were saying, there's the manufacturing elements and then also considering the logistics around it and how the field has just been getting more used to dealing with these things. So having experienced service providers that can also deal with transporting the cells to and from the hospital sites to get it to the central manufacturing location, having a lot of these CDMOs be more in tune with what it takes to do a cell therapy, have the right processes in place. All of these things have really streamlined it a lot more these days, whereas in the earlier days, it was a very new thing, even to some of these major CDMOs to implement it. So it involved a lot more effort to get it going. So I think these kinds of things have gotten a lot more standardized, and I think it's helped reduce some of the cost and complexity involved with doing the cell therapies for the field as a whole. I think the area that has still had a lot of constraints on it, and obviously with Squeeze we're biased, is the flexibility of what you can engineer about the cells has still remained relatively constrained. So when you think about people moving towards generation of cell therapies, that we don't do these kinds of therapies, but let's say CAR Ts with an edited PD-1, the process is still you're going to use a virus to insert the CAR, and then you're going to do something like electroporation to insert the CRISPR element to do the editing. So these things haven't been streamlined, and you're going to compound complexity once you start to try to go after anything more sophisticated biologically. So moving forward, it would be great to be able to adopt newer generation techniques that can actually make the process of engineering these cells much, much simpler. And then related to that, starting to integrate the process of manufacturing cells into single units or these pods that others work on, et cetera, that can allow you to have a closed system, ideally end to end. Because once you can achieve that, now you can get rid of the constraints of the clean room, et cetera, and really relax what it takes to implement the cell therapy and allow the turnaround times and accessibility of these things to become much, much more practical for broad implementation.
Great. So Armon, you kind of preempted what I was going to ask next, which was around bottlenecks in cell therapy. So you mentioned a couple of those things regarding the engineering of multiple components within cells. Are there other bottlenecks that you'd want to highlight within the cell therapy space that you think need to be addressed or should be addressed for the basic continuous advancement? You can start with
I
Armon,
think
if you have anything else to add, and then Kinnari, and then Ed.
Yeah. No, I think for a lot of the bottlenecks in the cell therapy world right now, if you have a new cell therapy that you want to get going, if it's virus-based, I think everyone's familiar with the kind of notorious wait times you would need to deal with to actually get those viral systems produced and have them produced at a GMP scale that you can use for your therapeutics. So just implementing a new cell therapy can take a long time because certain things like viruses can be a big bottleneck. And increasingly, I think the FDA is getting more sensitive to how do you think about these modifications? When do you think about stuff like potency assays? What are the long-term consequences? Like they came out with the guidance on you need to track gene-modified therapies for about 15 years or so. So there's more constraints that have been added to the system. And when you think about logistics outside of kind of the manufacturing and company side, but on the clinical sites, cell therapies are very involved trials from a clinical site perspective. And in an environment where they have staffing issues like many others might, it makes cell therapy trials that much less friendly and desirable to deal with. When a biologic is very simple, but a cell therapy, they have a lot more logistics and complexity to deal with. And they also can have infrastructure issues where if you're reliant on leukapheresis, for example, for your product, which many cell therapies that are autologous would require leukapheresis, that can be a limitation within that site because they have other things they need to use those leukapheresis machines for. So there's many different steps. There's some that are logistical on the site side, like I mentioned. There's some that are on the manufacturing side. But all of these things one would need to get better and better at addressing these bottlenecks to try to really bring forward these newer cell therapies.
I agree with what you said, and I think maybe I'll expand from a patient perspective, right? COVID has been enormously unexpected challenge, right? When you think about ex vivo lenti, where you need to get patients to the sites and sites that are qualified to do cell and gene therapy, apheresis and cell collection, there was a period, I think about 6 to 8 months, where the borders were closed. So Rocket had to work with embassies at different countries to get an exemption for patients to come to the clinical site for initial treatment. And that worked, but follow-up visits, it was a challenge, right? So I think the COVID situation has made 2 different bottlenecks we weren't expecting, is just getting patients to the site in clinical studies, especially in ex vivo lenti. And the second one is, I'll reemphasize, it's really the logistics. When the shipping and the number of flights from one site to the other gets decreased to 1/3 of what it used to be, it makes a bit of a challenge for cells to get from one place to the other. Analytics. So I think one of the challenges Rocket had is we were worried that our samples may get lost or stuck on our tarmat somewhere. So we ended up getting dual samplings and doing some things at the site-specific level so we don't lose the data. And I think those challenges were unique to the entire field of cell and gene therapy. And I think we've learned from each other and shared knowledge how to come past that. And I think some of that becomes a little bit normalized. The other challenge was follow-up visits that I talked about for patients. What we ended up doing is having nursing services that can go to different patients' homes and to collect the data so you don't lose the follow-up visits data that you need in order to really justify the benefit risk. And speaking of benefit risk, the other, I wouldn't say bottleneck, but it kind of is, right? When we talk about talent and resources for us as a company, that's also a challenge with our health authorities. So FDA is so under-resourced that a lot of the meetings you used to have in person due to COVID, they're remote. But even those meetings, they're not able to support because they're just understaffed and they're handling 3x more INDs they ever expected compared to the number of headcounts they have. So having that frequent, ongoing, transparent dialogue with the agency that I'm a huge advocate for along the ways, it has become a little more challenging. And I think one of the benefits we have at Rocket is because we have 4 programs and 2 with RMAT designations, we're able to use one program's advantage of learning and it apply to the other programs. But if we only had one therapy that had key milestones every 2 to 3 years, I think that would have been a bottleneck for developing a drug more efficiently. So health authority, logistics, patient trials, we're all learning. And the more we share knowledge amongst each other, I think more we can de-risk these challenges as an entire industry.
Yeah, I think, Armon and Kinnari have touched on some of the really key themes that again, at Achilles we've experienced over the last 2 or 3 years, whether it's, the kind of logistics or challenges at clinical sites. I think due to COVID, I think one of the other things, touching on the number of clinical trials that are ongoing, and specifically in oncology in the area that we're interested in, is again at clinical sites, the competition for patients, the competition for resources at the clinical sites. It's not always possible for some of these big centres to entertain, huge numbers of industry sponsors for their studies. And so, again, when we think about patient selection and getting the right patients onto your trials, I think that there is more than one thing to choose from for a treating physician now. So I think that's a challenge to procure enough patient samples to kind of, meet the timelines of delivering studies and data readouts. So something that I think a lot of people have been challenged with. I think it's definitely fair to say that it's not just perhaps in cell therapy, but perhaps something we're seeing more acutely is the challenge with kind of regulatory review times and getting people on the phone or kind of face-to-face meetings to talk through CMC sections or IND submissions. And I actually think that we're seeing the bar go up for cell and gene therapies. I think for a long time, perhaps because there was a feeling, that they were moving away from this academic environment. I think, the bar or the lens is being shone a little bit more on cell and gene therapy and more kind of leveled against what we've seen with traditional biologics and pharmaceutical manufacturing. So I think, that's just something that we're evolving as a kind of community and beginning to understand expectations from regulators before we put submissions in, trying to predict, what some of the questions that will come back, whether it's safety testing, potency testing, that I'm sure we're going to talk about as well. But I think, again, on the kind of footprint piece, the challenge of getting into CDMOs, if that's what you choose to do, is, there is a long list of people queuing at the door to get in, especially for kind of virus and vector manufacturing. And I think that when you're in a CDMO, it's how do you scale up to deliver more doses to patients as well? There's an acute shortage of space, and so we're seeing more and more companies commit earlier to their own manufacturing facilities to take full control of that. But then that again comes with significant capital investment ahead of clinical data. So it's a constant trade-off of trying to get ahead of the curve and deliver patients samples and doses earlier with not necessarily having kind of a huge clinical data set behind you. But those are some of the key themes that I think that we see on a day-to-day basis at Achilles.
Ed, so you touched on a couple of things there in terms of what regulators are looking for and whether the bar is shifting, in particular with regards to potency and release assays. Those are things that have been a topic of conversation in gene therapy and cell therapy in a number of different arenas. So can you discuss what that looks like for your modality and your understanding of what regulators are looking for at this front, both at early stages as well as at later stages of development? And Ed, we can start from you and then go to Armon and then Kinnari.
Yeah, and I think if you just kind of take a step back to say, maybe not quite 10 years ago, but 5 years ago, the idea that you'd be thinking about how to validate a potency assay at phase 1, phase 2 was not really in people's mindsets. It always felt like something I'll tackle that when I actually need to at late stage and into commercial. So I think, again, developers now are being pushed much earlier to think about what the assay needs to look like, even ahead of starting your clinical studies. And that's no different for Achilles. I think being able to understand what your active drug component is, especially for TIL-based therapy, what are the targets is absolutely critical. And at Achilles, because we are able to prospectively identify our targets for each patient's tumor by the bioinformatics kind of platform and algorithms we use, it enables us to use a potency assay it consists of the patient's clonal neoantigen peptides that are used in the manufacturing process. And that enables us to do two things. A, characterize the product. We understand what is the active drug product or drug component, and then measure that. And we do that through looking at cytokines, interferon gamma and TNF-alpha in an intracellular cytokine release assay. So firstly, we understand what is the active drug component. It's then understanding how do you track that in the patient. And so when you've got a kind of platform assay with the one that we use, we can look at the, tracking those cells and watching them expand, in the patient after dosing, and being able to then look at, that data in concert with clinical data is quite powerful when you think about, the expectations over being able to demonstrate efficacy and what's driving the mechanism of action. I think beyond that is having a matrix of approach for potency. So it's not relying heavily on one assay, because I think the regulators are going to steer us much more towards being able to look at a variety of different assays to demonstrate we understand the characteristics of the product. You might choose only to take one through to kind of a commercial product. You want it to be as easy and simple as possible. But the data set that you have to rely on, I think now, to being able to demonstrate you understand, what the product is doing in the patient is going to be absolutely key. And so we've, again, at Achilles, very early on, before we started our clinical studies, made sure that we built out a very robust number of assays that we wanted to look at outside of the release of the products, with regards to kind of for information-only testing and looking at things like, the kind of TCR sequencing of those drug products to understand the kind of makeup of that, and then also looking at a variety of different cellular markers, fitness, and as well as the phenotype. So it's about collecting a big data set early on that enables you to kind of define, what is the assay that you want to take forward as you move through the clinical development pathway.
Yeah, and I think just to add to those components, the FDA probably started with a kind of much more let's learn together stance with cell therapies early on, because so much was unknown, and why over constrain a new innovation before you know what you need to or don't need to be concerned about. And I think now with a lot more cell therapies that are in trials and having a lot more experience with it, it's getting a little bit more specific on what are the concerns and what are the things to think about. I think the potency assay elements that Ed brought up is a great example where early on it felt like they don't necessarily care about it in the early stages, but now you kind of have to discuss it in the early stages just to make sure you're on the right track for the future. And then I think they've gotten more in tune with what are the things to worry about and not worry about, both from a manufacturing perspective and from a clinical development perspective. So what are the sources of toxicity, et cetera, to be concerned about? How do you think about dose levels for these things and adjust for that? But given the focus here is more on the manufacturing side, thinking about how do you validate some of the genetic alterations that you've done? What do you need to be conscious of for long-term potential side effects of that? And how do you think about allogeneic versus autologous things differently? Because allogeneic therapies are going to have certain elements about them that you might need to be concerned about, which you wouldn't have been concerned about with an autologous therapy. For better or for worse, allogeneics, oftentimes you also have a lot more time to be doing your release testing, because an autologous thing tends to have a clock. Someone's waiting to get their treatment. Allogeneic, you're creating a batch. It doesn't inherently matter if your release takes a month, or at least as far as a regulator would be concerned. So you can actually become more exacting on an allogeneic format because you have time to look into it. And I think all of these things evolve, and probably the preference will tend towards, maybe to state the obvious, more predictable and simple outcomes. So for example, like taking it to the kinds of stuff we do with Squeeze, it's a vector-free system. It's just a mechanical disruption. So that eases up a lot of concerns on what is the vector doing, how did you produce the vector, et cetera, et cetera. It's just irrelevant. So they'll only think about the cargo you're putting in. So whether it's a peptide or an mRNA or what have you, in that case, they're much more used to regulating those compositions, and it becomes simpler. And I think they've kind of gotten a feel for how do you think about things like viability or other factors that might matter. Where do you draw the line for what those need to be? So as the experience has improved, I think it streamlines certain things, but on the other hand, results and requirements on certain areas that they might feel they have their hands around and now want to start setting some standards.
Yeah, I think the analytics, what we've learned over the last few years is invest in your analytics team early as possible, even before you submit an IND, right? Potency assay, but other release testing, in-process testing. I think health authorities have become more knowledgeable, more savvy, and it does, the more data that you generate and have an internal database of, the more you can justify and understand and rationalize what data you're seeing and why. I think that scientific evolution of understanding is changing. I think for us at Rocket, what we've done is the inventors of our therapies, we have them, some of them we've actually hired into as Rocket team members when we brought their programs on board. Others, we keep them very closely related to us, because talking to KOLs and scientific experts that really understand the vector design, the cell processing, the analytics, what are the functional assays that you really need to distinguish something for one disease versus another disease, taking them to health authority meeting, I found is also valuable to justify why you're picking what release assays you're picking and why certain things have worked. And while the guidance may want you to do X, Y, and Z, it's not feasible in some of the diseases to do it. And I think the one thing I do appreciate is while the health authorities have evolved their thinking as they learn more, they are becoming more consistent across and rational across how they think through things. So I think learning how to position what you have based on data to make justifications early as possible is great. And that allows us to just do things a little more efficiently and know early as possible when do you lock down. Specifically for AAV, what I'll say is regulators are thinking early as possible, lock down your cell line, right? Or lock down your process. And the sooner you can do that, the more you can utilize your patient level data as well as product level data to justify a smaller number of studies or number of patients per study in order to make your BLA MAA package more robust and consistent in its interpretation. So I think one of the learnings that we've had at Rocket is where possible, we try to have a commercial grade product in place from patient 1 onwards. And if not, at least by the time we approach a phase 2 pivotal study, we lock down everything and centralize all of our analytics and testing and stability data, et cetera, in order to get the database you need. Because even some of the newer guidances that the agency is working on right now requires that you need about 3 patients worth of stability data to be available, right? And in these rare diseases that we all work on, patient material it's like gold. You barely get access to it. And when you have access, how do you prioritize what you're going to use that patient data for? You may not necessarily, hey, stability is not the best place to use that patient material, that you have very limited access. Maybe you want to use it for potency or comparability. And I think those are the discussions we're starting to have with health authority as well as our scientific community. And I think there are some ways that we can improve and rebalance the pendulum that's swinging on one side to be a little bit in the middle.
Great. So in a lot of your answers to the first question in terms of innovations and how the field has changed, I think you guys mentioned things like various enabling technologies and analytics that have enabled better understanding of what's in a cell therapy product. So I'd like to expand on that a little bit. For example, technologies in single cell sequencing, proteomics, various omics capabilities have really increased the ability, at least in the academic side, of understanding what's in a complex mixture of cells. To what extent can things like that and other enabling technologies further refine the cell therapy manufacturing process development in terms of increasing the understanding of what's in a cell therapy product? And we can start with Armon and then Kinnari and then Ed.
I think that is, like you said from the beginning, going to be one of the areas that can really help advance the field. And I think right now the core of cell therapy is coming down to, okay, you want to make a cell do something. How do you make it do it? And the critical step there tends to be you're trying to insert some kind of material or gene, et cetera, that's going to have some downstream effect. And that is how you're going to make the cells go and do that. And if you can make those processes more flexible than they are right now, you can start to have access to a much broader range of biology that you can now engineer into these cells. I think right now for a lot of things, you can essentially add one thing at a time when you go and use viral systems. That's more or less what they do. They'll go genetically integrate a particular gene of interest. If you try to do 2 things, it gets significantly harder. If you try to do 3 things, it's way harder than that. And then if you want to delete things, you have CRISPR style options where you can deliver that component, then you can delete things. And if you're trying to do both of those, you need to put these kinds of processes back to back. So it's something where it's relatively inflexible to go beyond like single modifications, essentially. And that's something that, again, with the bias of squeeze, if you can really open that up and say, whether you want to do 1 thing or 6 things, you can just go do it all at once because you have a much more flexible delivery system. Now you can actually start to get much more sophisticated with what kind of engineering you can do in the cell and really advance by leaps and bounds on what's possible biologically with these cells. And by the way, also get into the zone of what if you did mostly transient modifications to these cells instead of permanent modifications? Because there's many things you can probably accomplish just by having done a transient modification. And then you don't need to worry about the long-term side effects. Or you can go after something very potent because you're not going to keep it on in perpetuity. You're only going to keep it on for a defined period of time. Like I think an illustration of that is the recent paper with someone who had CAR Ts still detectable, I think it was 10 years or something after their first administration. So on the one hand, yes, that's fascinating. On the other hand, if that person has CAR Ts 10 years later, that means they don't have B cells 10 years later because the CAR T's job is to kill off any B cells that exist. And these are the kinds of things that if cell therapies can become more nuanced and the range of functions they can go after, et cetera, that can really expand what the field can do down the line. So I think that's one major metric for how do you expand the biology of what cell therapies can do and have it become applicable to broader ranges of therapies and also come to earlier settings. Because what you'll tolerate from a tox and long-term issues perspective for a last line oncology patient is very, very different from what you would do from a first-line oncology patient. And when you're getting into non-life-threatening diseases, it's a completely different game. No one is going to be okay with CRS for, an autoimmune disease or something like that. The second domain, I think, would be more and more automation and integration of unit ops. So the more you can condense down what that manufacturing chain looks like, you can make it simpler, you can reduce the number of operators, you can remove the probability of error for different things. And that can also really make these things much more cost-effective and accessible. The ideal being, of course, you have integrated system that can do everything and can therefore sit at the hospital site, but any steps in that direction will always help the practicality of the cell therapies.
Yeah. I love the innovation that's coming about, right? I think about all innovations as what does it do? What does it help us de-risk and understand, right? And I think to increase the understanding of when you're talking about ex vivo lenti type of products or CAR Ts, individualized patient products, when there's variations that you have on the final product or what's made of it, I think decreasing or predicting what the variabilities mean and how to predict it from an efficacy and safety perspective, the more we know at a cell level, the better we'll be, right? So a lot of times VCNs, when they were a few years ago thrown out, right? People were trying to compare VCN to VCN, but it really is the methodology behind how you do VCN. So a bulk VCN versus CFC VCN versus the newest thing that's going on in characterizing analytics is single cell analysis of the lentiviral integration allows you to have a better predictability at a cell level of what that VCN or transfection could look like. And that can help us understand better what's the efficacy and predictability of safety for that product from both efficacy and safety perspective. I think the advances being made by, our scientists in the field are great. And I'm looking forward to ASGCT coming up to read more about this. It's not mainstream enough to be incorporated right now into programs that are already in the clinical studies and late stage that we are in. But I think this will just help us get into a very where instead of working on life-threatening diseases where any kind of VCN predictability, because patients have no therapeutic option and you can predict benefit risk, it's still worth it, right? That potential risk that you have 5-10 years down the road, if my LAD1 patients, 2/3 of them die by the age of 2, if they live 10 years down the road and they have a safety concern secondary, that to me, I've gotten drugs in oncology approved for 3-year extension of survival, let alone 10 years, right? So the benefit risk here is worth it. But as you take these cell and gene therapy products to more of the generalized medicine, bigger populations, I think these type of specificity in analytics, characterization, and scalability will allow us to do this in a way that makes it even more tangible for more normal diseases that are chronic, but not life-threatening.
Yeah. I think from my perspective, it's a super exciting time at the moment with the kind of innovation and technology that enables us to understand exactly what's happening within a patient's culture of their T cells. And so, again, if you think of when you're very early in development, the driver is to get your process into clinic as fast as you can. And we know that the kind of commitment to look at automation very early on is challenging. So the elixir for us really is just to be able to, in an autologous cell therapy space, is to be able to determine exactly what the feeding regimen or the cytokine concentrations are for each patient's process that enables us to ensure we get reproducibility. We're always hitting a certain dose threshold for each patient. And again, I think the technology is just starting to catch up. So if I think some of the things that we're interested in very much is the sensor technology that is starting to come into the space, being able to measure glucose and lactate and other kind of cytokines and metabolites that will enable us to take a more sophisticated approach to how we feed and harvest our patient cultures, so that inline testing, being able to understand what is the cell concentration and what does the kind of expansion kinetics look like. And I think very early on you're often just a little bit blind to that because you're kind of wedded to some of the technology that perhaps doesn't have that innovation. What we have done at Achilles though is kind of prospectively start to collect samples inside our kind of clinical manufacturing and development space, because we know at some stage we'd like to go back to those samples, because it might help us to characterize the product, to understand what's happening in those cultures, without necessarily having the technology readily available to us. So I think it's advancing, kind of at rapid speed, and things like 2D kind of fluorescence spectrophotometry and Raman mass spec are also kind of different technologies that we're looking to explore from an inline testing perspective to understand, how do we understand more what's happening with our autologous cell therapies.
Got it. Great. So in the last few minutes, I want to turn to some of the more operational logistics issues regarding cell therapies. In some of your earlier answers regarding the bottlenecks, I think each of you touched on some of the challenges relating to collection of patient material, for dealing with logistics at the administration site. So I'd like to expand a little bit on that and how some of those challenges can be addressed to make cell therapy more widely available and commercially feasible. So, for example, things like addressing the cost drivers, things like addressing global scale up or scale out, as the case may be in cell therapies, and just a wider reach. So, any closing comments from each of your perspectives on what can be done to alleviate some of those challenges? And you can go from Armon to Kinnari to again.
I guess maybe one way to think about it is just like walk through the whole cycle of getting material from the patient and then bringing it back to them. So if you start with, I'm a patient who wants a cell therapy and I go in, what kind of cell therapy is it? If it's allogeneic, we don't even have this upfront component. But if it's autologous, if it's a leukapheresis, we need to worry about chair time for all of that. If it's a blood draw, that's simpler, and then ultimately this material needs to get shipped. Now, often shipping is fresh because it's complicated and tricky to make sure the sites are all freezing things properly. So you have a very time-sensitive upfront logistics chain for that. PBMCs tend to be more time-sensitive than red blood cells. So something that's a red blood cell autologous thing, you have flexibility on how quickly you get it to your manufacturing site. If it's PBMCs, you better be getting it there in 24 hours or less, typically. Once you're at the manufacturing site, the big cost drivers are essentially time and operators and any materials that might be expensive. So vector can be very expensive, but otherwise clean room time and operators is by far the biggest factor. If your process takes a month, it's going to probably be an order of magnitude more expensive than a process that takes a day. And then for going back, I think if you can have a frozen material, that is a lot simpler because you're much more flexible on logistics of getting it back to the patient. If you're crossing borders, you have more time for customs, et cetera. And then once it gets back to the patient, then the question becomes, in some ways, what was the biology of what you did? Did they need to have gotten preconditioned? Are they going to need an ICU bed or some dedicated facility to stay in because they're going to have terrible side effects from this? And that's going to be one burden from a patient and system perspective to deal with. But if it's not going to require any of those and it's an outpatient thing, then it's a much simpler and more amenable piece. Because I think as we think about beyond the efficacy and safety of this for patients, when we think about the cost of the system, the rest of this chain matters. Like if it's a therapy that's going to hospitalize someone for 2 weeks, that is a much bigger burden to the system than one that could have been an outpatient setting. So I think these are all the factors that people need to consider as they go into it, and all the cell therapies that are around right now might be better or worse on some of these dimensions.
I think one thing I'll advocate for is work on more therapies across the platforms, right? You can have a lot of synergies. We have 4 programs and a wave 2 programs. Whether it's ex vivo lenti or AV, there are synergies that you can negotiate and optimize from vector analytics. Even when you think about plasmids, right? 3 versus 4 systems, there are optimizations that you could do and synergies you could have. So from a CMC manufacturing perspective, what we've tried to do for our programs is where possible, we have 2 or 3 programs in the same CDMO that's commercial, a great CDMO that's already de-risked by FDA AMA inspections, having those from the beginning, and I know that is hard to do, investing in that even before a phase 1 study startup really helps alleviate comparability or risk of slowing down the program or needing additional clinical studies. I think that scalability of, even if you don't have scale in number of patients you're treating, the centralization from a logistics perspective help. From a clinical site perspective, it's similar to what's been said. What we try to do is center of excellence. So we picked clinical studies of center of excellence where UCL Gosh, for example, in UK, as I look at our London-based panelists here, they have 3 of our programs that they're clinical site for. So when we do synergies and training for their team members, it's across all of our programs. And that allows us to really gain efficiencies. And also, the one thing that makes, I think, the biggest difference is knowing your ecosystem, whether it's your CDMOs or academic institutions or hospitals and clinical. The more you can collaborate and partner, the better it is. When you come to the logistics challenges, telling your PI, hey, there's an issue with this and we have to delay a patient treatment by a week, right? When you have a great relationship and they know the why of what we need to do to reshuffle, and there's constant reshuffling that's needed. So investing in great project managers and alliance managers that can reshuffle and refinagle things as things move around is always great. Where possible, also having not single source dependencies or single vendor dependencies, always another way to de-risk ourselves. So when you have logistics, having two vendors that can actually carry your products and be validated to do that allows you flexibility. So when one is having a challenge to a certain region, you can always rely on the other. I think those are the things we thought about. And the other component that is really helpful when you're thinking about developing a therapy in not just US or not just Europe, but both regions, having a decentralization or having a manufacturing facility or self-processing facility or analytics in each of the regions allows you to de-risk the issues when you have to ship samples across, right? So that de-risk that is so valuable because this patient material, this drug product, it's too valuable to get lost in some logistics component. And I think those are some of the things we're thinking about. But as we share more information, we learn from others of what we need to incorporate for future programs as well.
Not sure how we're doing for time, but maybe I'll just kind of summarize some of the previous points that have been made. And I think one that I guess is an intent focus for us is that Kinnari pointed to is like access to patient material ahead of either generating data for INDs and IMPDs. So it's in really tough and rare disease space, very tough when you need a patient's tumor to be able to demonstrate that you can validate a process. And so again, thinking prospectively about how are you going to de-risk both your manufacturing process and showing that you can validate that process, but also de-risk the supply chain and engage with clinical sites. And so, Achilles, we opened up a kind of non-clinical material acquisition platform that gave us access to a patient's tumor and their blood at our clinical sites. So these are patients who are consenting to give their material, but they don't get treated with a product. They're simply giving their material, for the benefit of Achilles and being able to establish our processes and procedures. And so early engagement with clinical sites so that when you do go into clinical studies, the teams know how to procure material, they know how to ship it appropriately and temperature controlled and the documentation that goes with that, all the way through to shipping to our manufacturing site and then back the other way when we think about final drug products and being cryopreserved. So, being able to do that in multiple different indications, so, opening it up into, different tumor indications has enabled us to kind of do early R&D and development work ahead of kind of committing to commencing clinical studies. And I think that's hugely valuable for us. But I know that, we've relied very heavily on in this field on either tumor cell lines or patient healthy apheresis that don't represent what real life is like inside clinical studies, and especially when you're accessing patients who have already undergone 3 or 4 lines of therapy. And that's certainly an apheresis, a patient that's had 2 or 3 lines of chemotherapy does not look like a healthy donor apheresis that you can buy commercially. So it's understanding how you de-risk some of those things before you get into clinical studies. And I think that those are some of the key things that we often look at.
Great, fantastic. So think we're at the end of our session, so I really appreciate all of you and your insights regarding cell therapy, and thanks for participating and for the great discussion that we've had.
Thanks a lot for having us.
Thank you so much.
Many thanks.